Mesothelioma is an aggressive form of cancer that develops from the mesothelium, which is a protective lining that covers most of the body's internal organs. The mesothelium lining the lungs, the pleura, is the most commonly affected area. Most cases of mesothelioma are caused by exposure to asbestos. Currently, diagnosis of mesothelioma relies primarily on medical imaging, pathology examination of biopsy samples, and cytology evaluation of pleural effusions. However, liquid biopsies offer a minimally invasive method to aid early detection by analyzing biomarkers circulating in body fluids like blood. Several biomarkers have shown promise for detecting mesothelioma in liquid biopsy samples. One of the most studied classes of biomarkers are circulating tumor cells, CTCs. CTCs are cancer cells that have shed from the primary tumor into the bloodstream. Their presence in blood correlates with worse prognoses in several cancer types. For mesothelioma, the detection of CTCs has been associated with advanced disease stage and poorer overall survival rates. Several studies have highlighted the potential of capturing and analyzing CTCs from blood as a liquid biopsy approach for mesothelioma. Techniques like microfiltration and immunomagnetic enrichment have been able to successfully isolate CTCs in blood samples from mesothelioma patients. Molecular characterization of the isolated CTCs through techniques such as immunostaining, fluorescent in situ hybridization, and gene expression profiling has shown that they exhibit epithelial and mesothelial markers characteristic of mesothelioma tumor cells. The presence of 3 or more CTCs per 7.5 milliliters of blood was found to correlate with poorer overall survival in one study of 50 mesothelioma patients. Larger validation studies are still needed, but CTC analysis shows promise as a liquid biopsy for early detection and prognostic monitoring of mesothelioma. Besides CTCs, cell-free nucleic acids like circulating tumor DNA, CDNA, and microRNAs circulating in blood are also being investigated as promising liquid biopsy biomarkers for mesothelioma. CDNA refers to fragmented DNA from tumor cells that enter the bloodstream during tumor progression and metastasis. Studies have detected CDNA in blood samples of mesothelioma patients and shown that CDNA levels correlate with tumor burden and response to treatment. One challenge is that CDNA concentrations can be very low, even in late-stage cancers. However, advances in techniques like digital PCR and sequencing allow for highly sensitive detection down to a few tumor-derived DNA molecules. Specific molecular alterations like BAP1 mutations that are commonly found in mesothelioma tumors have also been detected in CDNA of patients. Serial CDNA monitoring holds potential for non-invasively tracking treatment response and detecting relapse. MicroRNAs are small non-coding RNA molecules that help regulate genes. Various microRNA profiles have been identified that show deregulation in mesothelioma tumors compared to normal tissues. Studies have begun profiling microRNA signatures in blood samples and exploring their potential as mesothelioma biomarkers. MicroRNAs like miR25-3P have been found at elevated levels in blood of mesothelioma patients compared to controls, suggesting they could serve as liquid biopsy biomarkers. Another promising class of biomarkers are exosomes, nano-sized extracellular vesicles actively secreted by tumor cells into biofluids. Exosomes contain biomolecules like proteins, DNA, RNA and lipids that reflect the status and phenotype of their parent cell. They are increasingly being investigated as liquid biopsies that can provide a minimally invasive window into the molecular profiles of tumors. Studies have shown mesothelioma cells secrete distinct exosomal microRNA and protein profiles compared to non-cancerous mesothelial cells. These tumor-derived exosomes and their cargo can be isolated from body fluids like blood, pleural effusions, and urine of patients using techniques like differential ultracentrifugation. Mesothelioma-associated exosomal proteins and mRNAs identified in these studies demonstrate potential as non-invasive imaging and diagnostic biomarkers. Ongoing research aims to further validate exosomal biomarkers and develop targeted assays for clinical mesothelioma detection and characterization applications. In addition to the various nucleic acid biomarkers discussed above, studies are beginning to identify promising mesothelioma-associated protein biomarkers measurable in liquid biopsies. For example, mesothelin is a cell surface glycoprotein highly expressed by mesothelioma tumors as well as some other cancer types. Elevated levels of soluble mesothelin and related peptides have been detected in blood and pleural effusions of mesothelioma patients, showing potential for disease monitoring. Other glycoproteins deregulated in mesothelioma tumors and measurable in biofluids include osteopontin, fibulin 3, and fibronectin. Large-scale proteomic profiling studies continue to uncover additional candidate protein biomarkers with potential clinical utility, alone or in combination, for non-invasive mesothelioma detection through liquid biopsy platforms. Liquid biopsy analysis leveraging a wide array of biomolecular markers, including CTCs, KDNA, microRNAs, exosomes, and proteins show significant promise as minimally invasive tools to aid early detection, characterization and management of mesothelioma. Ongoing research aims to further validate promising biomarker signatures, develop platforms to efficiently capture and analyze target analytes at sufficient precision and scale in clinical settings. Combining results from multiple markers and assay modalities may ultimately enable robust, non-invasive liquid biopsy-based diagnostic and prognostic testing to improve mesothelioma patient outcomes.